Good morning, friends. So today it gives immense feeling that I should discuss a very interesting and relevant to the society. That is a topic on discard to diagnosis. The whole idea of discard to diagnosis, and specifically that should be affordable and accessible platform for rural, remote areas, inaccessible areas, specifically in the context of like India and the large country or developing countries, we can say. And these affordable and accessible avenues, specifically for early diagnosis of inherited metabolic disorders, that is abbreviated as IMD. I am Professor Nilesh Kumar Sharma at DPU Pune, India. So we should begin with the whole idea why there is a need of discard to diagnosis, specifically of IMD, inherited metabolic disorders. This one. So, my dear friend, in a very short and brief, IMD is also referred as inborn error of metabolisms, IEM. And in fact, IMD or IEM, it collectively refers a group of clinical diseases where specific enzymes are non-functional, specific enzymes and their counterpart genes are mutated. That leads to the failure of those enzymes to work so that certain substrate is not going to be converted into the product. That means it is the non-functionality of the enzyme, and that is why it is referred as inborn error of metabolism. And why it is inherited metabolic disorders? Because metabolic disorders are there, but those metabolic disorders are actually inherited from mother or father, and most of the cases it is autosomal recessive genetic conditions. Okay, so what is the need here? We know, and there are already knowledge is available, what are IMD, what are IEM. So what is the need here? That is diagnosis. The need is here management of IMD, and mostly these IEM and IMD that starts clinically develop from the newborn stage till the uh, adulthood stage, you can say. So some of the IEM or IMD, they are reflected at the early stage, and some little bit it takes time so that clinically symptoms are detected. So what is the whole idea is the early diagnosis of the IEM and the IMD is very critical. And why it is critical? So these are here is the point for the better management of clinically severe and moderate risk IEM. We can manage. So see, the cure of the disease is very difficult and it is less available, but we can manage the IEM or IMD by the proper external management like dietary management, nutritional management, etc. So the early diagnosis can also help the prevention of severe clinical symptoms. If it is early diagnosis, if you are managing the metabolic diseases or disorder at early, so the severe clinical symptoms can also be minimized, and that is why the mortality and the morbidity among the newborn or the children can also be reduced. That is the need. Another aspect is that like dietary management, nutritional management can help besides the expensive gene enzyme therapy or some other replacement therapy. So, so what are the avenues are available for the early diagnosis or the diagnosis of IEM or IMD? We know there are certain genetic screenings are available for selective IEM. Those are very well known, but there are many IM collectively, those are not so frequently known. That is one of the gap is there. So in spite of the genetic screening for certain selective IM, there is a gap in the early diagnosis of the IMD or IM. Another aspect is that there are certain approaches in terms of biochemical tests are available, but these biochemical tests are also selective. Only for few IEM are available for the early diagnosis. So what is the, and another very important aspect is this selective nature of diagnosis, whether genetic or biochemical, is not enough for the large population. And also very important to mention that the diversity of IEM or IMD are increasing across the globe, whether in the Indian setting or other continents. Why it is? Because the environments are changing, like pollution, nutrition, diet, chemicals, industrial waste, water contamination, air contamination. So all together, it is changing the landscape of the the severity of IEM, the types of IEM, those were not known earlier, now it is going to be clinically reflected. So this change in the environmental aspect also contributing to the IMD or the IEM. And that is why the need is to go for comprehensive, wider diagnostic approach. And that is why the whole idea is for today, that is that discard to diagnosis, metabopsy. Metabopsy, as I have already discussed in previous discussion, that metabopsy is a summarized terminology for metabolites based biopsy, and that is why it is abbreviated as metabopsy. But the metabopsy in the context of discard to diagnosis is very important here, and that is why this discussion I'm going to highlight. So what is the discard concept here? So let me to go to the next slide. So what is the whole discard concept? Any biological materials or the tissue, those are routinely, knowingly or unknowingly discarded from the human body. Like, for example, I say milk teeth is one of the discarded material, knowingly or unknowingly. And another is that is the nail clippings. And specifically, I will come to the point, first nail clipping is very important. Other than the discard nature of these biological materials, another very important point is, like milk teeth, the development of milk teeth during the gestation period or the pregnancy period starts from the 10th to 12th week. That means the whole nature and the chemical or the chemistry of the milk teeth actually relates to the from the mother to the offspring or the newborn. That is what the connectivity. That means the milk teeth is going to represent both chemistry inherited from the mother and also to the newborn. So it is like a bridging point. Another very important point is that is the first nail clipping. This nail clipping, the nail clipping development also starts during the pregnancy period, that is from 6th to the 8th week of the gestation period. So both milk teeth as well as the first nail clipping is the representative biological material. Those are routinely discarded, knowingly or unknowingly, represents certain chemicals, certain metabolites, certain clue for inborn error of metabolism, because inborn error or IMD is a sort of inherited metabolic disorders. From the parents to the offspring or the newborn, it is going to be transferred. So it is highly logical to, to understand the metabopsy of milk teeth and also the metabopsy of first nail clippings of newborn. And that may give certain idea about the whole discard to the diagnosis for the inborn error of metabolism or the IMD as a early diagnostic. No doubt genetic screening is there, no doubt other biochemical tests are there. Those are selective in nature. So this approach of the discard to diagnosis will serve as a collective, comprehensive early diagnostic uh, avenues for inborn error of metabolism or the metabolic disorder, inherited metabolic disorders, so that it can be early diagnosed, and so that the better management of these IMD can be fulfilled to the it is like less well known among the society, less you can say seriously taken by the health agencies or society or the family or the parents. So it is also a sensitization. How can we uh, employ these discard to diagnosis approach for the early diagnosis, so that the newborn or the children, those will be, if they have any IEM or the IMD, whether clinically severe or the moderate or it is not so severe, can be better managed as early as possible. And this discard to diagnosis, specifically from the milk teeth metabopsy and first nail clipping metabopsy, will be driven by our own in-house VTG tool, where we are intending to utilize those milk teeth lysate or the first nail clipping lysate by a novel and IP protected methodology for the diagnostic approaches. Thank you very much. And please be connected and keep listening to this Metabopsy channel. Thank you very much.